So the pathogenesis of atherosclerosis, this is really important, is understanding the disease process, because you'll have no hope of properly identifying the root causes to address if you don't understand the pathology. So basically, the traditional model, or the orthodox model, powerfully held, is that the LDL particles come from your bloodstream in the lumen of your artery, and they get into your arterial wall from the inside out. Okay? And lots of orthodox people in the business would say that that is the way it is. But there's an alternative view. Is it possible that the LDL particles come from the outside in? Now that would be really groundbreaking. And that's the view held and published on by Professor Vladimir Subotin. And we'll go through some of the details now of the hypothesis. So a short history of cholesterol, very brief. We had Anichkov way back in 1913, and the belief was that cholesterol interacted very much with atherosclerosis. And in the 70s and 80s, there was a lot of debates. We had a lot of crazy stuff came out in Time magazine and elsewhere. And then the NIH decided they'd push a consensus and stop the debate. So what was agreed was the elevation of blood cholesterol levels is the major cause of coronary artery disease. And there was solidarity around that, and any dissenting voices were squashed. Now, another key thing in the publications over the following decades was the absolute certainty that lipids, or LDL particles, enter the arterial wall directly from the arterial lumen. So this is the foundation stone of the whole thing. And again, there was solidarity around that, and there still is. It's a dogma that can't be challenged, but we'll challenge it today. So the LDL pathway of getting into your arterial wall is in many, many publications. This is a hugely well-known one, much cited. And basically, the progression of atherosclerosis is shown quite simply through from LDL getting into the intima, your inner wall of your artery, through to it building up, immune system getting involved, foam cells, and then you've got a kind of a hot mess of immune reaction. So that's accepted. But important to note is that there's a simple dogma that the LDL particles come in to the intima, the inner layer of the artery. So let's look a little closer at that. So looking a little closer at the morphology, we see an artery here, and on the very inner layer is the endothelium, a single cell layer of endothelial cells. Then we've got the intima, right? And that's shown as usually as a thin layer. Then we've got the media, shown usually as a thick layer with smooth muscle cells. And then the adventitia, which we won't talk about too much. But if you look closely at a cross-section, again, we see the intima shown thin, the media thicker, and the adventitia. But for very small arteries, this picture is pretty true. However, for large and coronary arteries, it's very, very different. So we see the small intima here in a tiny artery. Actually, it's very large in a coronary or a major artery. And the relatively big media in a small artery is actually relatively smaller than the intima in a larger coronary artery. And Subotin noted that the design, or this design, of coronary arteries pertains to all humans without any exception. And that's correct. So this is really important. I'll just briefly show some more of Subotin's slides. A 7-day-old here has a very thin intima, really just an endothelium, and the media is relatively much thicker. At 5 years old, however, the intima is thickening, naturally and physiologically. At 15 years old, the intima is almost as thick as the media. See the media here? This is the intima. And at 30 years old even, the intima is actually bigger than the media. And that's the way it is. It's a natural process where the intima gets to be a dense cellular compartment that grows much bigger than the media is. The differentiation of coronary intima into multicellular layered compartment occurs in all humans without exception. Right. Now, there's many publications that show this, no shortage, and they show physiologic diffuse intimal thickening, DIT. It's physiologic, it's normal. But the leading publication on the cholesterol hypothesis, which is Hansen 2005, and all papers practically, present a completely incorrect design. They generally show a thin intima which is mostly empty. And it makes it appear that, yeah, LDL particles could get in there. Interlopers. Nearly all of the big publications on the cholesterol theory show this incorrect view. That's the way it is. So why? Why would they all be incorrect on fundamental pathology and morphology of arteries? Seems weird. And Subotin suggests that the reason is quite simple. A depiction of the real human coronary morphology is not suitable for the cholesterol hypothesis. Because if you go through the actual real morphology, questions naturally arise, which we'll touch on now. And it goes against the concept that lipids enter the arterial wall directly from the arterial lumen, or the center where the blood flows. If you look at the proper morphology, that seems unlikely. So that's his belief that all of the diagrams are wrong because it's easier that way and it fits better with the dogma. So we're going to show you now where the cholesterol actually builds up in the intima in your artery. And you may be quite interested in this. And we'll show a visual view, which is from kind of a light microscope, an LDL or lipid view where they've stained for LDLs, right? So you can see where they are, and an immune cell view, which we won't concentrate on too much. So basically in a healthy artery, there's your big thick intima. And when you look at it stained for lipid or LDLs, there's none in there, right? So there's no real red staining. We've got our LDLs here, and we're going to see where they end up when disease progresses. Very early lipid buildup then, you see that the red staining for LDL or lipids is right down at the bottom of the intima. See this red area? So it's not building up anywhere else. So if they were coming in from the intima, you'd wonder why they're not all throughout, or even more concentrated here on their way down. But they're not. Increased lipid buildup, you see this red band right down at the media interface. So again, it's all building up at the base of the intima. Excessive lipid then, when you're at a more advanced stage, you've got all of this hot mess going on here. And pathological intimal thickening, now you can see that there's pockets appearing everywhere. And you're also seeing macrophage are moving in and trying to deal with the problem. And here's pathological intimal thickening with macrophage and foam cells. So you can see here all the lipid is filled up the intima and you've got foam cells all over the place, right? But it gathered originally in its earliest stage down at the base of the intima, far from the blood flow, down at the base near the media. So many publications show what I just told you, that this is the way the lipids build up. So this is a fact. Subotin and his outside-in model, how could this be true, you would wonder. So we'll quickly go through it. So here shows another cross-section, and I'm going to introduce the vasa vasorum, or effectively the arteries of the arteries. So thick-walled coronary arteries need to be nourished. They're nourished from nutrients from the blood that diffuse into the intima, small molecules mainly. And then they're nourished, the artery wall, by vasa vasorum, which penetrate into the media from the outside. So they're nourished from both sides. But I want you to watch something. When the intima is insulted, and this can occur from many, many root causes that I often talk about, the vasa vasorum are sent signals to come in and vascularize or to feed deeper into the wall, right, to help. And they can penetrate into the intima, the base layers of the intima. And they can here easily bring in cholesterol, LDL particles. In fact, where they could bring in the particles is kind of exactly where we see it in real life slides, as I talked to earlier. The vasa vasorum are a perfectly reasonable mechanism to bring in the LDL, an alternative to the cholesterol hypothesis. Atherosclerotic coronary arteries in many publications fully accept that the atherosclerotic coronary arteries have way more vasa vasorum and they penetrate all the way in. This is a classic kind of artifact of atherosclerosis, and normal arteries have very relatively few vasa vasorum and they don't penetrate. So this is in the literature. And we have many papers which show that the vasa vasorum penetrating are an enormous part of progressing atherosclerosis. So it's in the literature. So let's look now briefly. Serious progression towards coronary events or heart attacks and or death. Subotin would say that vasa vasorum or vasa vasora are an enormous part of this. And the orthodoxy would also have to acknowledge that vasa vasorum outside in are an enormous part of the real progression to serious atherosclerosis and death. So they both agree on that. However, in the early initiating step, it's where they disagree. So the orthodoxy would say that LDL particles in the lumen, where the blood flows on the inside, they make their way to the endothelium, to the intima, and then they jump and end up building up, as I showed you, at the base of the intima. So that's the orthodox view, and it's very rarely discussed as to exactly how that happens, though there are mathematical models, but very few serious proofs. Subotin, however, in his hypothesis, would say that many insults like hyperinsulinemia, diabetes, oxidative stress, yada yada yada, hypertension, they insult the intimal layer. And the intimal layer is extremely hyperplagic. In other words, when it's insulted, its cells will rapidly multiply and it will respond. And it sends signals to bring in vasa vasorum, to bring in neovascularization of new blood vessels to come in and nourish the intima that's in trouble. And of course, if you look closely here, this is where LDL particles would be introduced into the base of the intima, right? Which is what we happen to see in real life slides. So this is Subotin's hypothesis. For the orthodoxy, and here's an important distinction between the two hypotheses. For the orthodox hypothesis, the dominant root cause is higher LDL particle numbers, more invasion from the lumen, right? So that's what it says to us, that it's all about the LDL particles. Whereas Subotin, if you follow his hypothesis, it's all about the dominant root causes being the intimal insult, right? Which is not LDL. It's hyperinsulinemia, oxidative stress, many kinds of rare diseases that cause vasculitis or inflammation, smoking, all the real root causes of heart disease, I would say, right? So this is a big difference. We got to emphasize my classic list, the main root causes. So the evidence for and against, and then we'll go on to the interview with Vladimir. So the evidence for the orthodox hypothesis is that the LDL can cross the endothelium. We know that from some papers I went through in talks last year. But transcytosis, which is the main process and brings LDLs across, why would evolution design that process in order to give us atherosclerosis? Transcytosis, bringing in LDL, has lots of other reasons, but not giving us atherosclerosis, surely. LDL can cross, yes, but leaky junctions, another way that LDLs can come across, they're due to root causes. So leaky junctions are caused by our list of root causes. So it's not really LDL's fault if you've got leaky junctions between your endothelial cells because, you're doing other things wrongly. LDL could traverse the intima, could make its way across, in principle, but the entrapment is only in the deepest layers. And why isn't there LDL seen throughout the intima in the earlier stages? LDL could maybe traverse the intima, but so far I've mainly only seen mathematical models and diffusion models, not actual proofs, but we're looking for those. Higher LDL could exacerbate the general heart disease problem, but high LDL particles can also exacerbate the problem in Subotin's model, because if you have more LDL particles here and you've caused damage to your intima and you're bringing in, blood components and LDLs, more will mean it'll be worse. So this does not distinguish or prove the orthodox hypothesis. And we could say that biglycans, you'll hear that in the interview, the biglycan structures are more concentrated in the deep intima, so the LDLs could come across and only really get stuck where there's biglycan. But there's contradictions in the literature about that, showing biglycan in lots of other areas of the intima, where again the LDL don't build up. So, this is debatable. Briefly, we'll look at Suboten's evidence for his hypothesis, and we'd say that all the mechanisms are supportive. But we would say that vasovasorum bringing in the early LDL to the base of the intima is not actually proven yet. It is a hypothesis. Root causes drive the intimal insult that brings in vasovasorum. That's pretty established. So that's 4. Intimal insult drives biglycan. So literature shows that the insult to the intima drives the generation of the biglycans that trap. So again, that's something in support of Suboten. Hyperplasia leads angiogenesis. So briefly there, hyperplasia or cell growth of an insulted intima has been shown in some studies to be 3 weeks ahead of the vasovasorum coming in. So again, the sequence in time supports Suboten. The vasovasorum tips at the very tip as they come in. At the end, are fenestrated or leaky, so they could very admirably allow these particles to come in. Outside in precedes endothelial issues. So other studies show that the vasovasorum are coming in before there's actual endothelial distress seen in certain studies. So again, it's supportive. And I'll skip that one. Perivascular adipose. So perivascular adipose tissue is the fat that's around your coronary arteries. And there's other papers that show that this fat is required to be present to have a problem. And this is an outside-in phenomenon. And vasovasorum are required for significant atherosclerosis. Wherever you have atherosclerosis, you're going to have vasovasorum, right? So certainly at the scene of the crime, always. And corneal vascularization data. So the study of the eye has shown that when you get lipids in the eye in certain disease states, that it's always preceded by arteries growing in, neovascularization into the eye structure before you get a buildup of cholesterol. So it's kind of a proof point from a different area of the body. The strong focal nature of atherosclerosis. So atherosclerosis occurs badly in specific spots in the artery, right? And nearby there might be none. So it's very focal, whereas leaking through the endothelium of LDL particles would be a more general thing. And HDL efflux via the lymphatic system. So we now know that the HDL removing cholesterol from the wall actually takes it all out via the outside, right? So it's another proof point. A lot of people don't know that. HDL gathers cholesterol and takes it out via the lymphatic system, which is like vasovasorum, and takes it out to the outside of the artery. So there's no inside out there either. So